This is a short video going over the steps for a muscle contraction. So for a muscle contraction to occur, first we need, at least in a skeletal muscle, a nerve signal to reach that muscle cell. Neurotransmitters have to go across that junction, and that's going to open up the passageway for calcium ions to go through. So the process there is called depolarization. You'll learn more about that in Bio 30. But calcium ions are stored in the sarcoplasmic reticulum. The sarcoplasmic reticulum lets those calcium ions out. They're going to go in amongst the myofilaments, and they're going to stick. So these little bits here are calcium ions. They're going to stick to troponin. This little one here is troponin, and troponin is going to do a twist. That twist will allow tropomyosin, this thinner, in this picture, blue rope protein, is going to get out of the way. Right now it's blocking the active sites on actin, but soon it won't be. As soon as troponin does its twist, tropomyosin gets out of the way, and now the myosin head can see that active site right there. Once that's possible, then myosin is going to stick to it. It really wants to stick to it. So the myosin head is going to, so here's the myosin head, is going to attach to that active site and do a pull. You might want to call it pulling, or the myosin head is bending, or that the muscle is contracting, or that actin is sliding past. The actin molecule is pulled in, which shortens the unit called the sarcomere. Sarcomere has a border here and a border here. That's that striation you can see under a microscope. This pull requires the use of ATP, and later you're going to need ATP to get all of these calcium ions, back out to the sarcoplasmic reticulum. So you need ATP both for the contraction and to get rid of calcium ions at the end. This is a myogram, and a myogram shows the tension in a muscle tissue. And at the beginning, there is a period, if we have a stimulation that happens here, there's a period called the latent period where it seems like nothing is going on. But what's happening there is it takes time for the calcium ions to attach to troponin to get out of the way, tropomyosin to expose those binding sites, and finally myosin can stick to actin and do its pull. So when myosin is attached to actin and pulling, that's the contraction phase, and the relaxation phase is when those calcium ions are pumped back to the sarcoplasmic reticulum. For our types of muscle tissue, we've got 3 in our bodies: smooth, skeletal, and cardiac muscle tissue. Smooth muscle is this one here. Smooth muscle is shaped in a tapered kind of way, narrow at the ends, fatter in the middle, and we find it in our esophagus, in our arteries, we find it in our organs. Our cardiac muscle is having a branched kind of shape. Those branches allow it to connect to lots of neighbors and share cytoplasm between neighboring cells. That's important because our cardiac muscle is not allowed to take a break. We have to keep our heart beating. The skeletal muscle is tubular in shape. It has often many nuclei and many mitochondria. Both the skeletal muscle and the cardiac muscle are striated. They're stripy. If you just look at the stripes under the microscope, you can see that. Smooth muscle is not. Your skeletal muscle tissue might have lots of myofilaments. Remember, these are the clusters of actin, myosin, troponin, and tropomyosin. The sarcoplasmic reticulum wrapped all around, containing the calcium ions. And in around there are nuclei that can direct the repair and creation of new myofilaments, and also lots of mitochondria to provide the ATP to make all this happen. Of course, that means we also need to have, depending on our function of the muscle, but if it's a slow twitch muscle anyway, we have lots of oxygen stored nearby in myoglobin, and almost all of our muscle cells, so this is for all of them, have lots of sugar stored nearby in the form of glycogen. Because remember, you can use glycogen to do both your aerobic cellular respiration, which uses oxygen, and your anaerobic cellular respiration, which does not require oxygen, but it's less efficient, although it's a bit faster.